talk now about how all of this is regulated. So we've got enzymes in our body, right? We've got the GI enzymes, for example. We've got the DNA enzymes. We've got lots of enzymes across different body systems and so on. And so they're always existing in the presence of product as well as the presence of substrate. So there must be some mechanism to actually control the rate of these reactions, to turn these enzymes on and then turn them off. If you think about it, we don't always want to be breaking down fats, right? There are enzymes that break down fats like lipase, enzymes that break down proteins like protease. We don't always want these enzymes to be active. Let's say we are not let's say we didn't just have a meal, there's no food in the GI, we're in a fasted state, then we don't want these enzymes turned on. So we want them turned off. And so regulation of enzyme activity is how we turn enzymes off in the absence of substrate or product, depending on the direction, or we can turn them on again, depending on the concentration of substrate or product. So let's look at the 4 mechanisms that are involved in regulating enzyme activity. So the first mechanism is called the allosteric regulation. And allosteric regulation is simply involving a modulator. So there's a modulator of some sort. This can be X molecule that binds to a regulatory site on that enzyme. So we've got 2 sites here. We've got the regulatory site and we've got the active site. Remember, the active site binds the substrate, but the regulatory site binds some other molecule. But this molecule is going to regulate the enzyme's activity. It's going to alter the enzyme's activity depending on some other factor. So the modulator could be sensitive to maybe substrate concentration, maybe product concentration, maybe some other chemical factor, and it's going to turn the enzyme on if it's a favorable outcome, if that's what's going to be a favorable outcome depending on that factor. And it can also turn the enzyme off if that would be a favorable outcome as well. So some modulator enzyme, some modulator molecule X will bind to the regulatory site of this enzyme, and it will alter the enzyme's activity depending on what would be a favorable outcome for the situation in which the enzyme is in, okay? A second mechanism here is called covalent regulation. And covalent regulation is slightly similar, but this talks about some chemical group. This is usually a phosphate group. It can be other things. It can be sulfur as well, but basically some type of chemical group binds onto this enzyme, again, not at the active site. This is the spot for the substrate, but somewhere else on the enzyme. And then another enzyme helps to catalyze the formation between covalent bonds, which adds this functional group on. That will help the reaction to proceed in one direction. And then if we remove this functional group, which is done by another enzyme, enzyme B, that will help the reaction to proceed in the opposite direction. So this can be either turning the enzyme on or off, depending on what enzyme. But basically we've got one enzyme, enzyme A, which is not the enzyme in question. Okay, so let's make that clear. So enzyme A is not the enzyme that we're actually regulating. It's another enzyme that's involved, but all it's doing is it's adding this functional group, which is usually a phosphate, via a covalent bond to the enzyme that we're actually regulating. This could turn the enzyme on, make it go faster. This could also turn the enzyme off and make it go slower. And then this can happen in the reverse direction. We have another enzyme that comes in, it cleaves this covalent bond, removes this chemical group, and then we've got the chemical group and the enzyme. And in this scenario, again, this could be turning the enzyme on or off depending on the specific enzyme in question. Okay, so here we've got 3 players involved. We've got the enzyme that's actually being regulated, turned on or off. And then we've got 2 other enzymes that are either adding a chemical group, such as a phosphate, or removing a chemical group, such as a phosphate. And the presence or absence of that phosphate will determine whether or not the enzyme is active or inactive, depending on that specific enzyme. okay? And so in this way, we have other enzymes, enzyme A and B, that are helping to regulate the enzyme in question by adding or removing that chemical group. This is a very common mechanism of regulation. We're going to talk about this specific mechanism again and again and again. When we get to muscle, we'll talk about how we can phosphorylate certain enzymes and turn them on, dephosphorylate them and turn them off. And so here's an example of that covalent regulation in action. So we've got the enzyme plus a phosphate. Let's say this is protein kinase. Protein kinase comes and takes that enzyme off. So we have the enzyme without the phosphate. And then on the other hand, we have a phosphatase, which can add the enzyme on. And again, we have the enzyme and phosphate on the other end. Okay, so let me explain that again. So we got the enzyme plus the phosphate. Protein kinase comes. If we think about a kinase is adding phosphates on. And we'll talk about examples again and again, but anything that adds phosphates is usually a kinase. There's different types of kinases that we'll encounter. And so we've got the enzyme and the phosphate group on this end. And then in the reverse direction, we've got another enzyme, which we can think of this as enzyme B from our previous example. This is coming around to cleave phosphate groups. And again, usually when we encounter phosphatases, they're cleaving phosphate groups off of the enzymes. And so now we end up with the enzyme and the phosphate group on the other end. Okay. And so this process is called phosphorylation. So we can phosphorylate the enzyme and then we can dephosphorylate the enzyme. And again, this will be an example that we use in many systems to explain how we turn certain enzymes on and off. All right. So let's see if there are any questions here briefly before we continue on. So any questions on the first two mechanisms of regulating enzymes? So allosteric regulation and then covalent regulation. All right, so let's keep going here. So the third mechanism is called feedback inhibition. This is where the rate of an enzyme is modulated depending on the concentration of a substrate that is upstream in the reaction. So a good example here is that in this reaction, we've got substrate A converted to substrate B, converted to substrate C, converted to substrate D, to product D, excuse me. And so we've got three enzymes in between that are catalyzing the reaction, and it's proceeding in one direction from A to D. Now, in this example, the levels of substrate C, the concentration of C, is what determines the rate of enzyme 2. So if we have a lot of C, then we want to kind of slow things down so that we're not producing as much of C and kind of decrease the buildup here. So what that does is that if we've got increased concentrations of C, then it turns off or decreases the activity of enzyme 2, okay? And so the rate limiting step here is going to be the conversion of B to C, because this is the step that determines the overall rate of the reaction. That's what we mean by the rate limiting step of the reaction. So I'll say that again one more time. We've got the conversion of A to B, B to C, C to D, and that is being catalyzed by three enzymes involved here. So enzyme 1, enzyme 2, enzyme 3. And the rate limiting step, the step that is determining the overall rate of the reaction, is the conversion of B to C via enzyme 2. So the concentration of enzyme C will determine the rate of enzyme 2. And that kind of makes sense, right? If we've got a lot of C building up, then we want to turn down or turn off the rate of C and decrease the amount of substrate or product C that starts to build up. Another example here is where the rates of E2 and E3 are constant in this example, but the levels of D, product D, will feed back upstream and alter the activity of enzyme 1. So this is another example of feedback inhibition. And so the conversion of A to B is going to be the rate limiting step in this reaction. This is the step that determines the entire rate of the reaction. So once again, if we have a lot of product D, then we decrease the weight of enzyme 1. And if we have a little bit of product D, then we can increase the weight of enzyme 1. So the rate limiting step will be the conversion of A to B via enzyme 1. And then lastly, we have a very rare form of regulation. This is called feed-forward activation. This is not very common in our physiology, but this is where the concentration of a substrate upstream affects the activity of an enzyme that's downstream. Again, very rare, but we do see it in some instances throughout the body. So we've got A converted to B. And so if we have a lot of product B, then we can increase or speed up the rate of enzyme 3. And that's just going to get the reaction moving faster downstream so that product B can be absorbed and converted to C, and so on and so on. So it's kind of just clearing the clutter downstream by speeding up the rate of enzyme 3 as a response to increased concentrations of product B. Okay. So I've got a question here. Is feedback inhibition similar to negative feedback? Yes, we can kind of think of feedback inhibition as being similar in some regard to negative feedback. The presence of a high concentration of a product will then go back and turn off the enzyme that's creating that product. So it is a form of negative feedback that we can kind of compare to, which is feedback inhibition, not feed-forward activation. Feed-forward activation would be more like positive feedback, okay? So we're actually increasing the rate of the enzyme upstream in a response to the concentration of a product or substrate that's upstream. Excuse me, the rate of the enzyme downstream in response to the concentration upstream, okay? Any questions about that? Feedback inhibition or feed-forward activation? And as you can see here, just like positive feedback, this is also very rare. And feedback inhibition or negative feedback is much more common than feed-forward activation. Okay, so I don't see any questions. All right, so let's wrap up here by looking at a few practice questions once again. Okay, so these questions are pertaining to enzymes. So let's kind of just put our knowledge to the test and answer these questions. So I've launched the poll. I'll give you a minute to poll in, and then we will discuss. Okay, final couple seconds here. All right, so the question asked if the activation energy of a reaction is high, then the rate of the reaction is. We had a lot of options for B, and B is correct, okay? And if you think back to that graph that we drew, the curve that had the higher or the larger activation energy is the curve that preceded the slowest. So if the activation energy is high, then that means that the reaction requires a larger amount of energy to proceed, and so it's going to proceed at a lower rate than if we had clear this, than if we had an enzyme present where we have a smaller activation energy. So remember, the activation energy is the energy that is required or consumed by the reaction in order for it to proceed. Okay, so I'm going to pause here. I got another interesting question. So the question is asking about covalent regulation. And the question is protein kinase a reactant as well as a phosphatase, or is it just what the reaction is called? Okay, so I'll go back to that slide here for a little bit. Okay, so covalent regulation is the name of the regulating mechanism. The enzyme that is being regulated is the enzyme in question. And then we have two other enzymes that are helping to do the regulation. Now, when we think of regulation, just think about what is turning the enzyme on or off so that it's not continually active. Regulation just means that we're fine-tuning the process so that an enzyme is only on when it's in use or when it's a favorable outcome, and then it's turned off when it's not a favorable outcome, okay? So the enzyme A and enzyme B can be thought of as protein kinase and phosphatases. These are just examples of enzymes A and enzyme B. And so what happens is enzyme A comes on, it's going to put this phosphate onto this enzyme that we're trying to regulate. And by putting that phosphate on, that could turn this enzyme on or turn this enzyme off. Okay, I'm not going to say that it's going to turn it on because you have some enzymes that are turned off by being phosphorylated. So it's going to change the activity of the enzyme by having that phosphate bound to it. And what does that? What does the binding of that phosphate is enzyme A, and an example of that is protein kinase, okay? On the other hand, if we want to reverse that change, we have another enzyme come in, which is enzyme B, an example of that being phosphatase. And what phosphatase does is it cleaves that phosphate off, and that will reverse the change of the enzyme, which will turn the enzyme off or on, depending on what that enzyme is. So it's going to reverse. If we turned it on by adding a phosphate, we're going to turn it off by taking the phosphate off. If we turn it off by adding the phosphate, we're going to turn it on by taking that phosphate off, okay? So in this way, in order to bring about covalent regulation, if you think about the word covalent, just means that the covalent binding of something, some kind of chemical group, is what is doing the regulating, and the enzymes involved, protein kinase or phosphatase, the examples here, are what are actually doing that covalent binding. All right, I hope that kind of cleared that up some. So the mechanism is covalent regulation. The enzyme in question is different from the enzymes that are helping to regulate, which are the protein kinase and phosphatase from this specific example. Okay, any other questions? Thanks for bringing that question up, because if you've got questions, it's likely that someone else has questions. Okay, excellent. Any other questions on enzyme activity or the four mechanisms of regulating enzymes? So make sure you understand like the big picture here. I know a lot of students tend to get kind of, caught up in the details of each of these mechanisms, but make sure that you're keeping the big picture in mind. What is the goal of regulating an enzyme, right? Making it turned on when it needs to be, turned off when it doesn't. So that's what regulation really means. So make sure you have a simplistic understanding of what the goal is, and then you can fill in the actual details of what the four mechanisms are, okay?